Complement cascade. A related group of sequentially interacting proteins called complement play an important role as an effector in both innate and adaptive immunity. Complement proteins are often activated by interactions with antigen-antibody complexes, but can also be activated by innate mechanisms in the absence of specific antibodies. Complement proteins can react sequentially to cause lysis of bacterial cells, or can label antigen-expressing cells for enhanced recognition by phagocytes, accelerating destruction of the antigens. Complement is made up of a number of proteins, many with enzymatic activities. These proteins are activated in a sequential, ordered fashion by antigen-antibody complexes on bacterial cells. The result is cell membrane damage, leakage of contents, and possibly lysis of the bacterial cell. Complement proteins are found at comparable levels in the serum of all individuals. A major function of antibody is to recognize invading cells and activate complement system for attack. Many antibodies, each specific for a single antigen, can recruit the ubiquitous complement proteins. The immune system does not need individual enzymes to attack each invading agent. The individual proteins of complement are designated C1, C2, C3, and so on. Classical activation of complement occurs only with antibodies of the IgG and IgM classes. When IgG or IgM antibodies bind antigens, especially on cell surface, the antibodies fix the ever-present complement proteins. The complement proteins react in a sequential cascade, with activation of one complement component leading to activation of the next, and so on. The key steps start with binding of antibody to antigens. Binding of C1 components including C1q, C1r, and C1s to the antigen-antibody complex, leading to C4, C2 binding at an adjacent membrane site, and activation and binding of C3. Membrane-bound C3 catalyzes formation of a C5, C6, C7 complex at a second membrane site, and C8, C9 are then deposited with the C5, C6, and C7 complex, resulting in membrane damage and cell lysis. The C5, C9 components are known as membrane attack complex because they interact at a single membrane site to form a pore. The byproducts of complement activation include chemoattractants called anaphylotoxins. They cause inflammatory reactions at the site of complement deposition. Reactions involving C3 result in chemotactic attraction and activation of phagocyte, resulting in increased phagocytosis. Reactions involving C5 lead to T cell attraction and cytokine release. Complement is a bactericidal and lytic agent for many gram-negative bacteria when used in conjunction with specific antibodies. However, gram-positive bacteria are not killed by complement, even in the presence of specific antibodies. Gram-positive bacteria can, however, be destroyed through opsonization. Thank you.